Hello everyone, I am Anis Lady and today I'm going to be talking a bit about how improve brain simulation accuracy, sensitivity analysis and realistic time delays in neural mass models. So this work started during my PhD with Pedro Valdez Sosa in China and is currently ongoing in Alan's lab at the MNI. And the rationale of this work is that there is a lack in general in theoretical formulation, particularly in the modularization and treatment of delays, especially in distributed delay that haven't been modeled until now. So with that, I would like to make a new interpretation for time delays and include the distribution of this transmission between neurons. So there is a need to improve the efficiency in algorithm, and for that I am going to be using some numerical integration that speed up the calculations. Sorry. So neural mass model should be also more realistic, and I think this is a very important topic because real data should be even more included in the brain modeling in general. So I am going to include some of the realism in the distribution of delays. So the hypothesis of this work was the make an efficiency of the neural mass model, and there are two key factors. One is the local generalization method for numerical integration, and the other one is the use of the connectome tensors. So the primary objective of this work is the advancing neural mass model using connectome tensors for high-dimensional data. So for that, we are going to make an integration of connectome tensors using neural mass modeling. Then I'm going to identify some of the previous limitations of existing modeling methods. Then make a simulation effects of the distributed delay on connectivity topology on brain dynamics. Then after that, I made an extension of the Janssen and Reed neural mass model, that is one of the well-known neural mass models, to include 1,000 of cortical columns. A sensitivity analysis of the model I am going to be also presenting, and as well I have developed some methods and a toolbox, free toolbox available already for high-dimensional neural mass models. So the brain modeling can be, let's say, divided depending on the level of description that you want to include on your analysis. So there is the microscopic level where you can explain how all the neurons interact. And in this type of models, they present like a very huge number of, let's say, differential equations that describe the process. And an example of that is the Blue Brain project. On the other hand, we have the mesoscopic level that is more like a collective behavior of neurons. And the mesoscopic level can be divided into neural field models and neural mass models. And in this work, we focus on neural mass models. So I use the Janssen and Reed neural mass model that contains 3 neural populations, 2 excitatory and 1 inhibitory. And for that, previously, I'm going to explain what do we understand for time delays. Time delays is just the time that neurons spend on transfer information between 2 regions of the brain. So these time delays is affected for different axonal properties, like the length, the diameter, the myelination, and the G ratio. And some researchers have treated the delays as instantaneous, which means no delays at all, or as a Dirac delta function, and which I think is not that realistic. So that's why we are presenting a new approach for that. So there are some researchers like Innocenti, who has proved that delays are very sensitive to the axon diameter. So taking this in consideration, I follow these equations for computing the conduction velocity. And for that, I need to previously calculate the G ratio and the diameter axon. So the G ratio is computed by DTI, can be extracted from that. It is just a relation between the myelin volume factor and the axonal volume factor. And the diameter axon has been proved for the Dirac-Schmidt that is very sensitive. It's a very sensitive parameters for the conduction velocity. So taking this in account, I follow the work from Sepperbank that he studied different type of probability densities to estimate the axonal diameter. And he found that the generalized stream value function distribution is the best fitter for the axonal diameter. So I took this distribution and I extract data from Caminiti and Lewald from diffusion tractography and electron microscopy, and I compute the conduction velocity. With that, I am able then to compute the time delays. And the length of the axons was extracted from diffusion tracing. So in this work, I introduced a new definition in neural mass model, and it's a connectome tensor. This is used instead of using a connectivity matrix. Why a connectome tensor? This tensor has two key components. The first one is the delay tensor, where all the time component is represented. And in this work, I explored three types of delay tensors. The first one is instantaneous delay, a Dirac delta function, and the distributed delay, that is a new approach incorporated in this work. Then the second component of the connectome tensor is the connectivity matrix. And for that, I also explore three types of connectivity topology. The nearest neighbor network, where just only local connections are included. The small world topology, where long-range connections are added. And the full connected graph. For numerical integration, I use the local linearization method that is a faster and more effective, let's say, numerical integrator for differential equation. And what happened with this method is that it preserves the dynamical properties of the system, and it is not explosive. And traditional methods like Runge-Kutta and Euler, they used to explode. So that's why I am using the local linearization method. There is an important thing that I want to highlight, and is that with a sufficiently small integration step, the input to each neural math can be assumed to be available. And this is used in this framework for accelerating the process. So yeah, as I said, I use the local linearization method for non-autonomous random stochastic differential equations. And these are the three key steps or key points of the method. Find good inputs to each neural math, and which will be already available, so you don't have to compute in each integration. And I have to use also I can use a very small integration step, and still getting good approximations. And this calculation can be also symbolically made, so which is a huge, let's say, effective thing for speeding the computation. So I also made a sensitivity analysis of the model that is basically based on the random forest algorithm. And I create a mesh of parameters using the Latin hypercube design. So now this sensitivity analysis was made for the steady state dynamics and the Seichert neural dynamics. And I'm going to show you later what are the results related to this aspect. So I took the Janssen and Reed neural mass model and I make an extension for analyze 1,000 of interconnected cortical columns. So let's say that the Janssen and Reed model is just for one single cortical column. So this is a very high dimensional problem, which could be even extended to a higher number of cortical columns. And in this case, you can interpret each cortical column for different brain regions. So imagine having, I don't know, interpreting or analyzing 1,000 of brain regions. So it's a very high dimensional problem. So I made some simulation changing the input to the model, and I found that when there is the mean above the threshold, the spectral dynamic show an alpha peak, and when we keep the mean and the standard deviation increase, the number of harmonics decrease, and for a mean below the threshold, a zeta rhythm appear. So we are proving that neural mass models are really sensitive to the inputs that we interpret, analyze, or include. So for that, I also analyzed how the extended Janssen and Reed neural mass model behave for different connectome tensors, let's say for different delay tensors, and for different connectome topology. So as we can see, for a Dirac delta function, we are having an alpha peak, and it doesn't matter which topology we analyze. And for the distributed delay, this rhythm shifts to a theta wave. And so we can conclude that from this part that the way of interpreting delays is very important in neural mass model, because we will get then different brain dynamics. So during the parameter sensitivity analysis, we found that, as many researchers have proved, that the neural mass model is highly sensitive to any variation on parameters, and the parameters related to the axonal properties, we also found very sensitivity. So as summarizing, we have presented like a general formulation of the neural mass model based on a concept of a connectome tensor. We also present and propose an efficient algorithm to integrate high-dimensional neural mass model based on our distributed delay connectome tensor. And the distributed delay were realistically calculated using anatomical data, and this was proved using previous results. And the effect of distributed delay showed that indeed the type of delay between neural masses can profoundly affect the dynamics of the model. And the extension of the Janssen and Reed was explored for different type of connectivity topologies and brain dynamics. So as well, the sensitivity analysis showed the most important parameters of the parameters for the steady state and seizure dynamics of the model. So that's all for my part. So now this is some work in progress. I am incorporating even more real data to, well, that's what I want, to incorporate real information, let's say electrophysiology, neurotransmitters, receptors to the model, and include also a specific layer information. So that's it. Thank you. More than welcome your questions.